Bismillahirrahmanirrahim. Dear students, have a nice day. Hope you will be fine best of your health. This is Dr. Sajid Mahmood Rao, Division of Science and Technology, University of Education, Lahore. Today we are talking about dihydrotestosterone, in which we will discuss about its classification, its functions and key mechanism regarding their synthesis. These are the key concepts which we will discuss in today's class session, and these are androgen receptors, binding affinity of dihydrotestosterone, function of dihydrotestosterone, metabolism of DHT, its properties and classifications for the same. Let's see what are the hormones and dihydrotestosterone is derived. Androgens are one of the main families of steroid hormones present in humans. Progesterones, androgens, estrogens, glucocorticoids, and mineralocorticoids. The androgens group consists of four hormones, that is dihydrotestosterone, testosterone, androstenedione, and dihydroepiandrosterone. The relative potencies of these androgens are 300%, 100%, 10%, and 5% respectively. The principal androgens are testosterone and androstenedione. They are of course present in much higher levels in men and play an important role in male traits and reproductive activity. Other androgens include dihydrotestosterone, that is abbreviated as DHT, dehydroepiandrosterone, that is abbreviated as DHEA, and dehydroepiandrosterone sulfate, that is abbreviated as DHEAS. The androgen receptor is a member of the nuclear receptor superfamily and binds both testosterone and dihydrotestosterone. These receptors reside in the cell cytoplasm bound to heat shock proteins until testosterone or dihydrotestosterone diffuse into the cell and bind the receptor, resulting in dissociations of the heat shock protein. DHT has a more potent androgenic effect than testosterone because its binding affinity to the androgen receptor is approximately double to that of testosterone, and moreover it has a dissociation rate about a fifth of that of testosterone. Let's see in the diagram how the dihydrotestosterone is derived from the concerned hormones. As you know, and you can see also over here, the testosterone is derived from the cholesterol. The difference between both the structure is that there is a presence of hydroxyl group at carbon 3 positions and a side carbon chain at 17 positions and a double bond at carbon 5 position in the cholesterol, while that of in testosterone the carbonyl group is present at carbon number 3 and hydroxyl group is present at carbon number 17. So in this way, from testosterone, the dihydrotestosterone is derived by the effect of enzyme 5-alpha reductase, and estradiol is also derived by the enzyme effect, that is P450 aromatase. In this way, the dihydrotestosterone is derived from the first intermediate which is synthesized from the cholesterol. Now come to the real topic, that is dihydrotestosterone. Dihydrotestosterone, that is also known as DHT, 5-alpha dihydrotestosterone, 5-alpha DHT, androstenedione or stenolone, is an endogenous androgen sex steroid and hormones. DHT is one of four principal androgens in humans and is synthesized primarily via the irreversible action of microsomal SRD5A, both types 1 and 2, on testosterone. DHT is the 5-alpha reduced metabolite of testosterone that is principally converted from testosterone in target organs such as prostate, skin, and liver. Synthesis can also occur from other precursors, but these pathways, although potentially important in some tissues, The common example is prostate, which is minor. Intracellular DHT is a more potent androgenic agonist than testosterone, and its presence in some tissues such as prostate is necessary for the full organ development and function. Circulating dihydrotestosterone levels are of much less important than testosterone for optimizing the intracellular DHT concentrations due to the presence of a rate-limiting enzyme, that is 5-alpha reductase, and is shown in SRD5A in types 1 and 2. Inhibition of these enzymes with 5-alpha reductase inhibitors decreases intertissues in dihydrotestosterone levels, and thus in certain tissues, that is prostate, that diminishes the agonist action of testosterone, thus reducing prostate size and function. You can see over here the basic skeleton of dihydrotestosterone, in which total number of carbon atoms are present 17, and the carbonyl group at carbon 3 position and hydroxyl group at carbon 17 position. Dihydrotestosterone is biologically important for sexual differentiation of the male genitalia during embryogenesis, maturations of the penis and scrotum at puberty, growth of facial body and pubic hair, and development and maintenance of the prostate gland and seminal vesicles. It is produced from the less potent testosterone by the enzyme 5-alpha reductase in selected tissues and is the primary androgens in the genitals, prostate glands, seminal vesicles, skins, and hair follicles. DHT signals mainly in an intracrine and paracrine manner in the tissues in which it is produced, playing only a minor role, if any, as a circulating endocrine hormones. Localizations of SRD5As in prostate tissues, that is type 2, skin type 1, liver type 1 and 2, and hair follicles, that is primarily type 1, catalyzes the formations of dihydrotestosterone from testosterone in these tissues. These enzymes expressed in the nucleus and cytoplasm of, for example, prostate epithelial cells are encoded by the 5-alpha reductase type 2, that is SRD5A2 gene, and polymorphism of this gene, leading to increased 5-alpha reductase activity and dihydrotestosterone concentrations in the prostate, that is have been hypothesized to increase risk of prostate cancer. The SRD5A3 gene has also been linked to increased dihydrotestosterone production in hormones refractory prostate cancer cells, and these genes may be particularly important in the metastatic prostate cells, which have been shown to express more SRD5A1 and SRD5A3 but significantly less SRD5A2. Conversion of testosterone to dihydrotestosterone via SRD5A activity in peripheral tissues is the main source of circulating DHT, but it is important to note that little DHT synthesized in the prostate or liver enters the general circulations due to efficient intracellular mechanisms that initially metabolize DHT to 3-alpha and 3-beta 17-beta androstenedione diol that have little androgens activity. As noted previously, DHT can also be synthesized in tissues by backdoor pathways that enable formations of DHT in the absence of testosterone or androstenedione as precursor. In yet a third synthetic pathway to dihydrotestosterone, namely the 5-alpha androstenedione pathway. 5-alpha androstenedione is converted by 17-beta hydroxysteroid B3 to DHT. As discussed later, these alternate synthetic pathways, which are not influenced by circulating dihydrotestosterone, may have particular clinical significance within prostate tissues. There are two isoforms of the 5-alpha reductase enzyme. Both are microsomal NADPH dependent proteins that reduce the double bond at the carbon 4 to 5 positions in a variety of carbon 19 and carbon 21 steroids basic skeleton. They differ in regard to their optimum pH, chromosomes locations, and affinity for testosterone. So you can see over here how the dihydrotestosterone is derived from testosterone by the effect of enzyme 5-alpha reductase 1 and 2, that is also abbreviated as SRD5A1, 2 and 3. This is the first step which is used for the synthesis of dihydrotestosterone. Let's see how different kinds of steroidal hormones are derived from dihydrotestosterone by the effect of relative enzymes. You can see over here in the second step 3-beta 17-beta androstenedione, which is a steroid which is derived from dihydrotestosterone by the effect HSD7, that is 3-beta DH. Similarly, the second steroidal hormones, that is 3-alpha 17-beta androstenedione, is derived by the effect of enzyme HSD5, 7 and 10, that is also known as 3-alpha DH. Both these enzymes are isomers of each other's because there is a difference only the positions of above and below the planes of 3-beta and 3-alpha of these steroidal hormones. After that, 3-alpha 17-beta androstenedione gives 3-alpha androstenedione glucuronide by the effect of enzyme UGT2, B7, B15 and B17, and also gives androstenedione by the effect of enzyme HSD2, 5 and 11. This enzyme finally gives androstenedione glucuronide by the effect of enzyme UGT2, B7, B15 and B17. The dihydrotestosterone also gives androstenedione by the effect of enzyme HSD2, 5, 8 and 10. Further, dihydrotestosterone gives DHT glucuronide by the effect of UGT2, B7, B15 and B17. Finally, the steroid 3-beta 17-beta androstenedione gives 3-beta androstenedione glucuronide by the effect of enzyme UGT2, B7 and B15. This is the basic diagrams and schematic pathway for the synthesis of dihydrotestosterone. Type 1 5-alpha reductase has an alkaline optimum pH of 8 and is active over a broad pH range, that is 6.0 to 8.5. The gene encoding type 1 5-alpha reductase, that is SRD5A1, is located on chromosomes 5 at PL 5.5, while that encoding the type 2 enzyme, that is abbreviated as SRD5A2, is located on chromosomes 2 at 2p23. The type 2 5-alpha reductase has at 10 to 15 fold higher affinity for testosterone than the type 1 enzyme. The type 2 5-alpha reductase has an acidic optimum pH of 5.5 and in contrast to the type 1 isoforms is only active over an extremely narrow pH range. The two enzymes are differentially expressed and regulated in the regard to tissues distributions and timing of expression throughout the life. The ontogeny of the 5-alpha reductase isoforms has been elegantly clarified by the author Thyfen et al., who have demonstrated the dynamic expressions in humans. The type 1 enzyme is not detectable in the fetus, appears transiently in newborn skin and scalp, and is permanently expressed in the skin, including pubic skin, only after puberty. There is no difference in type 1 expression between bleeding and non-bleeding scalp. The type 2 enzymes has been found to be transiently expressed in the newborn skin and scalp, the adult scalp, and pubic skin. Within the male reproductive organs, the type 2 enzymes has been localized to the testes, epididymis, and vas deferens, while type 1 appears more abundant in the seminal vesicles and ventral prostate. Let's see the comparative study of type 1 and type 2 5-alpha reductase isoenzymes, which is located in the tissues. In type 1, we can see over here it is transiently present in skin after birth, while type 2 skin transiently after birth. So in type 1 skin permanently after puberty, in type 2 pubic and scalp skin permanently after puberty. In type 1 axillary hair and follicles, in type 2 beard hairs and follicles. In type 1, it is present in liver, seminal vesicles, and ventral prostate, while type 2 is present in internal and external male genitalia and liver. The liver is the main source of circulating DHT in both genders, as it contains both type 1 and type 2 isoforms of 5-alpha reductase. The DHT found circulating in blood is predominantly from type 2 5-alpha reductase activity, that is 70 to 80%, with the remaining 20 to 30% from type 1 5-alpha reductase. Let's see the androgen receptor and its binding affinity of dihydrotestosterone. The androgen receptor is a member of the nuclear receptor superfamily and binds both testosterone and dihydrotestosterone. These receptors reside in the cell cytoplasm, bound to heat shock proteins until testosterone or DHT diffuse into the cell and bind the receptor, resulting in dissociations of the heat shock proteins. This enables the ligand androgen receptor complex to enter the nucleus, where it is phosphorylated and dimerized and binds to androgen response, that elements in the promoter regions of the androgens regulated genes after co-activator recurrent, thereby affecting genes transcriptions. DHT has a more potent androgenic effect than testosterone because its binding affinity to the androgen receptor is approximately double that testosterone's, and moreover, it has a dissociation rate about a fifth of that of testosterone. Organ differences in receptor binding of testosterone and dihydrotestosterone results in part from relative differences in intracellular concentrations of these androgens rather than from differences in receptor affinities alone. Indeed, it has been clearly demonstrated that high concentrations of intracellular testosterone can shift AR binding away from dihydrotestosterone by mass action. Moreover, despite there being a single AR physiological differences in testosterone and dihydrotestosterone action are well known and likely reflect variations in AR receptor distributions. Ligand-induced conformational changes to AR that affect stabilizations, local hormone synthesis and metabolism. AR ligand interactions with chromatin, cooperativity of the receptors with other transcription factors, and actions of co-activators and co-repressors. Thus, the local tissues controls of androgen levels in conjunction with numerous other factors driver AR-induced transcriptional responses. And as elucidated later in this lecture, tissues concentrations of androgens, particularly in the prostate, are partly distinct from circulating levels. Like testosterone, circulating dihydrotestosterone is principally bound to sex hormones binding globulin (SHBG) and more weak to albumin. In general, protein-bound dihydrotestosterone is inactive except in some reproductive tissues in which Magalin and endocytic receptor acts as a pathway for cellular uptake of dihydrotestosterone when bound to SHBG. Studies of interactions between a wide array of natural and synthetic androgens and SHBG indicate that the molecular structure of dihydrotestosterone favors tight linkage to the steroid binding site on SHBG. Compared with testosterone, dihydrotestosterone has roughly a five-fold greater binding affinity to SHBG. Binding of circulating dihydrotestosterone to SHBG in highest in young males, that is 0.5 to 2 years of age, 90%, and thereafter declines to about 70% at age 15 and to 40% in young adult men in that age of 18. The increase of SHBG that occurs with aging, that is approximately 1% per year, increases dihydrotestosterone bindings in older men. Dissociation rate constants from SHBG for dihydrotestosterone and testosterone have been measured in human serum and correspond to half times of dissociation of 43 dihydrotestosterone and 12 testosterone seconds, thus further demonstrating the tenacity to which dihydrotestosterone binds to SHBG. Accordingly, concentrations of free circulating dihydrotestosterone in gonadal men are very low and would be expected to remain so even when total dihydrotestosterone levels increase in response to TRT. This leads to an important question. Can an increase in circulating levels of SHBG dihydrotestosterone give rise to dihydrotestosterone mediated effects? Let's see the metabolism and history of dihydrotestosterone. DHT formed in peripheral tissues is extensively metabolized before its metabolites appear in the circulations. Metabolism of dihydrotestosterone to inactive steroids occur primarily via the initial actions of 3-alpha 17 beta hydroxysteroids dehydrogenase, that is 3-alpha HSD, and 3 beta 17 beta hydroxysteroid dehydrogenase, that is 3 beta HSD, in liver, intestine, skin, and androgen sensitive tissues. When we see the history of DHT, it was observed that dihydrotestosterone was first synthesized by Adolphe Butent and his colleagues in 1935. It was prepared via hydrogenations of testosterone, which had been discovered earlier that year. Dihydrotestosterone was introduced for medical use as an AS in 1953 and was noted to be more potent than testosterone but with reduced androgenicity. It was not elucidated to be an endogenous substance until 1956, when it was shown to be formed from testosterone in rat liver homogenates. In addition, the biological importance of dihydrotestosterone was not realized until the early 1960s, when it was found to be produced by 5-alpha reductase from circulating testosterone in target tissues like the prostate glands and seminal vesicles, and was found to be more potent than testosterone in bioassays. The biological functions of dihydrotestosterone in humans became much more clearly defined upon the discovery and characterizations of 5-alpha reductase type 2 deficiency in 1974. DHT was the last major sex hormone, the others being testosterone, estradiol, and progesterones, to be discovered, and is unique in that it is the only major sex hormone that functions principally as an intracrine and paracrine hormone rather than as an endocrine hormone. In 1968, Brock, Hoske, and Wilson published the first two papers describing dihydrotestosterone. Wilson and his colleagues subsequently demonstrated that 5-alpha reductions of testosterone to dihydrotestosterone was essential for the normal male fetal, genital, and prostatic development. As a result of this seminal work, dihydrotestosterone has been regarded as one of the three major sex steroid hormones in men, joining testosterone and estradiol. Now we will discuss dihydrotestosterone 5-AR 5-alpha reductase. Urologist Charles V. Huggins in 1901 to 1997 first applied the scientific method to the study of androgen-prostate relationships, earning him the Nobel Prize in 1966. The work Huygens performed in the late 1930s and early 1940s, along with his urological colleagues W. W. Scott and C. V. Hodges, has been deemed to the most important body of urological research in history. Now we will discuss the investigator and authors who have discovered and have been performed different kinds of work on dihydrotestosterone in different years. The first investigator is J. Hunter, that belongs to Scotland. In 1786, he discovered the prostate dependence on testes. The second investigator is Breithold in Germany. In 1849, he discovered the blood-borne nature of testosterone, that is first hormone. The next investigator or author is L. Rosica, that belongs to Switzerland, and A. Butent, that belongs to Germany. In 1939, they discovered the isolation synthesis of testosterone, and they got a Nobel Prize in 1939. The C. B. Huygens in 1941, that belongs to United States, he published responsiveness of prostate diseases to androgens deprivation therapy, and he got a Nobel Prize in 1966. J. Wilson in 1969 discovered the biological role of dihydrotestosterone. J. Ambrotto and McGinley in 1974 discovered the effects of congenital deficiency of 5-AR. The P. C. Walsh, that belongs to United States, in 1975, he discovered and co-discovery of 5-AR syndromes. The E. Stoner and G. Gomley in 1992 discovered the approval of finasteride. The R. Rittmaster C. Rohrbaugh, that belongs to United States, in 2002 discovered the approval of dutasteroids.